Okay, everybody, today we will talk about the infectious diseases. This is a very interesting topic. You know that most of the diseases occur due to infection by the microorganisms, right? So first we will talk about the microorganisms. Then we will talk about two types of cells or organisms. One is called prokaryotes and another eukaryotes. Then we will talk about the bacteria. We will talk about viruses, other microbes. Then we'll talk about antibiotics. That is the medicine that can kill the microorganisms. So we use antibiotics to treat the infectious diseases. We'll talk about antibiotics, then how the microorganisms spread in the nature. So first, you know that microorganisms, when we say microorganisms, when we say bacteria, right, people think bad things about it, right? Germ, like bacteria is everywhere and they try to harm us, right? We think common perception is that. But what is interesting is that there are many good bacteria that are essential for our life survivals. Okay, we don't think much about those bacteria. Inside the human body, this is a fact, but it is surprising fact is that in human body we have more bacteria than our own body cells. Okay, the number of bacteria inside the body is more than our own body cells. If you count the number in one human body cells, and we have 20 times more bacteria in the body. That means if you see the ratio, one human body cell, 20 bacteria. Okay, so 20 times more bacterial cells are present in the body. So you just think that our body is mostly formed by bacteria in that way, we can say. And although bacteria are much smaller than the cells, regular cells, so about 10% of your body mass is given by bacteria. That's a big, mass, right? So if your body weight is 150 pound, then it will be like 15 pound body weight is given by the bacteria inside your body. Okay, so it's amazingly true. Now, good bacteria and bad bacteria. So some microorganisms or bacteria are good, some are bad. Good bacteria includes the bacteria present in yogurt. Okay, inside your gut, intestine, you have huge amount of bacteria, okay, flora, and that help to digest the foods. Okay, inside your oral cavity, mouth, you have good bacteria, and they prevent the bad bacteria to infect inside your mouth. Okay, so we have good bacteria inside the oral cavity, so they are protecting us. So that is very interesting, right? Those microorganisms cause diseases. Those are called pathogens. So pathogens are the microorganisms that cause diseases. Okay. The cells could be two types. In the organisms or in the body, the cells could be two types. One type of cells are called prokaryotes. Another are eukaryotes. And eukaryotes. Okay. So eukaryotes are the cells present in the body of the animals, plants, and human, like multicellular organisms, right? Our body is multicellular, animal body multicellular, plants body multicellular. So eukaryotes are the cells in the organisms, multicellular organisms. So human body cell, for example, it has a nucleus, right, which is membrane bound, bounded by nuclear envelope, right, or nuclear membrane. We have mitochondria, we have Golgi apparatus, we have endoplasmic reticulum. All those organs have membranes. So membrane bound organelles are present in eukaryotes. Okay? Prokaryotes are unicellular, like single cell microorganism. Example, bacteria, only one cell, right? Single cell. And another difference is inside the prokaryote, the DNA are not present in the nucleus. No membrane bound nucleus is present. In eukaryotes, we know that DNA are inside the nucleus, right? Chromatins and DNA inside the nucleus. But in case of prokaryotes, DNA are present in the cytoplasm and not located inside the nucleus because no nucleus is there. So this is nucleus. Okay, and this is called nucleoid. Okay, nucleoid. No membrane bound nucleus, just DNA in coiled or circular form. So we can say DNA in circular form. Okay, in bacteria or prokaryotes. Inside the eukaryotes, the DNA are like strands, straight strands. Okay, so not circular, like straight strands. Okay. Another thing is many important organelles are missing in prokaryotes. Okay, for example, no mitochondria, no Golgi apparatus, no endoplasmic reticulum, but ribosomes are present in both. Okay, so these are ribosomes that produce protein. We know that. So these are ribosomes. Okay, are present in the cytoplasm. Cytoplasm. This is cytoplasm. Okay. So those are main differences between prokaryotes and eukaryotes. Okay. I'm repeating quickly again. Eukaryotes are the cells in multicellular organisms like human, animals, plants. Membrane-bound nucleus, DNA are straightened in strands. Other organelles are present, and most of those are membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi apparatus. Also in the cytoplasm we have ribosomes. In case of prokaryotes, no nuclear membrane, the DNA are in circular form, and that is called nucleoid. And in the cytoplasm, you have most of the organelles missing, ribosomes are present. Okay. Another thing is, if you see the cell membrane of an eukaryote and a prokaryote, there is a difference in the cell membrane. In prokaryotes, the cell membrane is stronger than eukaryotes. In the cell membrane, you have 3 layers. So, for example, in bacteria, you have 3 layers in the cell membrane. Okay, that is an advantage for the bacteria, right? Because bacteria are more protected. That's why antibiotic, it's difficult to kill bacteria because they are more protected. The covering is stronger, okay, tough. It has 3 layers. What are those 3 layers in the wall of bacteria? If you go from inside to outside, innermost layer is like exactly like this, the cell membrane or plasma membrane of human cell. Okay, probably you have studied in high school or intro-bio cell membrane, the structure of cell membrane. It is phospholipid bilayer. Do you know that? 2 layers of phospholipid. So, if you see the cell membrane of eukaryote, just one structure, and that is the cell membrane formed by phospholipid bilayer. That means 2 layers of phospholipid. This is one phospholipid, this is another. They form 2 layers. Okay, so this is the structure of the cell membrane. This one. Okay. In case of bacteria, if you see the bacterial wall, innermost is exactly same as eukaryote cell membrane. That means phospholipid. That is called the cell membrane, and then middle layer is called the wall, bacterial wall, and outermost is the capsule. So now you see the picture here. Cell membrane, also called cytoplasmic membrane, same as human body cell membrane, but additional membranes are the cell wall and capsule is the outermost one. Now, if you see the function of the nucleus, which is very important to understand how bacteria or virus work. So let's just briefly go over the function of DNA and RNA. You need to know to understand how bacteria or virus work. So RNA, ribonucleic acid, DNA, deoxyribonucleic acid. So those are very important components. If I draw a cell, okay, this is the nucleus. Inside the nucleus, you have a dense structure that is called nucleolus. Okay, so we are learning biology first. This is nucleus. Okay, and you have chromatins, thread-like structures. Okay, and chromatins are heavily packed in the nucleolus. That's why it is dark or deep color inside the nucleus. So chromatins are heavily packed inside the nucleolus, but chromatins are also present in the nucleoplasm. That means in the nucleus. Okay, now chromatins contain DNA. So just know that inside the nucleus we have DNA, and DNA store the genetic information, genetic material. Genetic information is stored in the DNA. What kind of protein your cell should produce? Okay, that is prescribed by DNA. That is prescribed by DNA. You know, you have many different types of cells, right? Gland cells produce hormone, one type of protein, right? And different glands produce different types of hormones, right? So DNA tells the cells to produce, okay, that kind of protein. So just know that that information is stored in the DNA. Okay. Now, inside the nucleolus, you have RNA. Okay. RNA are located inside the nucleolus. And ribosomes, you remember I said both eukaryotes and prokaryotes have ribosomes in the cytoplasm, very important. So let's see the ribosomes. Okay, so these are ribosomes. Okay? Ribosomes manufacture proteins. Just know that this is very important. Ribosomes manufacture what? Proteins. Okay? Now, I told you that nucleolus has RNA, and what type of RNA is called ribosomal RNA. Okay? Ribosomal RNA produce ribosome. Let me write down here. Produce. That's why called ribosomal RNA. Okay, ribosomal produced RNA produced ribosomes. Okay, so these ribosomal RNA here, they produce ribosome, and those ribosomes come out through the nuclear pore. We have pores or holes, so the ribosomes come out and enter into the cytoplasm. Many of them get attached to the rough ear, if you have heard that, rough endoplasmic reticulum. Most of them get attached to ear, some of them present in the cytoplasm. That's fine. So ribosomes are produced here in the nucleolus. Who produce that? Ribosomal RNA. That's why called ribosomal RNA, and they come out in the cytoplasm. Now, another type of RNA that are called messenger RNA. Okay, messenger RNA. And third type is called transfer RNA. So three types of RNA present in the cell: ribosomal RNA, messenger RNA, and transfer RNA. Make sense? Three types of RNA. Now, I have already mentioned ribosomal RNA. They produce what? Ribosomes. Make sense? And ribosomes come out and get attached to the endoplasmic reticulum or in the cytoplasm. And now you tell me, I have also mentioned, ribosomes manufacture what? Proteins, right? Now that ribosomes manufacture protein. Okay, good. Now, I said DNA. DNA stores what? Genetic information, right? Genetic information. That what kind of protein should be produced. Remember that? Okay. So, ribosomes produce protein, you know that. And DNA has the information what kind of protein should be produced. So, what happens? The DNA sends signal through another type of RNA that is called the messenger RNA to the ribosomes. Messenger RNA. Its name is telling you, messenger, right? Will bring the information to the manufacturer, right? DNA will give the prescription to messenger RNA, and messenger RNA will do what? Carry it to the manufacturer of protein. That means to the ribosomes. Make sense? So, messenger RNA takes the prescription or plan from the DNA to the ribosomes. And so, now, to just think about a factory, a factory that produces or manufactures the goods, you need what? You need the plan, right? That comes from DNA, what should be produced, right? What should be the shape, size of the goods. And then another thing you need, what is that? You need the materials, right? So, the materials for protein are what? Amino acids. Amino acids join together to form protein chains. So, amino acids. Right? These are amino acids. What happens? The amino acids get together, join together by peptide bonds. Peptide bonds. These are peptide bonds to form a protein chain, protein molecule. Okay? So, in the cytoplasm, you have amino acids. So, these are amino acids. Now, someone has to bring these raw materials of protein, that means amino acids, into the ribosomes, because ribosomes produce protein. So, who brings the amino acids from the cytoplasm to the ribosomes, to the factory? That is the transfer RNA. Okay, this one. So, those are three types of RNA. Make sense? And their function. Did you understand? So, if I ask you, which RNA produce ribosomes? RNA. Which RNA? rRNA, ribosomal RNA, okay? Its name is ribosomal RNA, produce ribosomes. And who brings the plan or prescription from DNA to ribosome? mRNA, messenger, right? And who brings the raw materials, amino acids, into the ribosomes? Transfer RNA. Make sense? Okay, so that's how the proteins are produced or manufactured in the cell. Now, why you need to know this, how protein is manufactured and processed in the cell? Because the microorganisms, many microorganisms, they multiply by producing the proteins inside the living cells, normal cells of the body. They enter into the cell and change the way the cell produce protein. So they start to produce the protein in their way, okay, inside the cell. So that's how they work. So this is very important to understand how the microorganisms work. Okay, so Now you know the difference between prokaryotes, eukaryotes, and why bacteria is so resistant against antibiotics or other medicines. Okay. We know the common microorganisms are bacteria, virus, fungi, right? Those are commonly heard. But prions, another type of, we will say prions are not microorganisms, but prions are proteins that cause disease. So prions are basically protein, no cellular components inside, no nucleus, no organelles, no cytoplasm, nothing. Okay? So these are not cells. Prions are not cells. Protein molecules. And these protein molecules are abnormal protein molecules. They get folded abnormally inside the cell. And that abnormal foldings of prions or proteins, these proteins, we say prion proteins. Prion proteins abnormally folded, and that causes diseases. The most of the time, prions cause diseases in the brain. Okay, encephalopathies. So prions are mainly responsible for any brain diseases, and that is called encephalopathies. Okay. We also say full name of these diseases, transmissible fungiform encephalopathies. Okay. TFE we often use. I'm sorry, here I see. Can you change this one? Transmissible spongiform. Spongiform encephalopathies. TSE. In shortcut, we say TSE. So, many different types of brain diseases can caused by prions. And those diseases together is called TSE. Now, one common of this probably you have heard. I have heard mad cow disease. Mad cow disease. This is one of TSE that caused by prions. Okay, and prions, as I said, are abnormally folded proteins, and they can also change normal proteins and make them prions. Abnormal foldings in normal proteins. Okay, and spread quickly and no treatment. So we don't know any effective way to fix that problem. Fix those proteins. Okay. Now, you already know the structure of a bacteria. Bacteria could be different types. We classify bacteria in two ways. One is we look at the shape of bacteria. Okay, by looking at the shape and by looking at the stain it takes. Okay, stain it takes. So based on the shape, we divide the bacteria into following types: cocci, bacilli, spirilla, and virus. So those are common types. Cocci, some people say cocci, are round shaped bacteria. Streptococci, strep throat. Bacilli, when we say I, that means plural. Okay? Bacilli are rod shaped, kind of rod shaped bacteria. Okay. Then spirilla are kind of spiral, not straight. Okay. And Hybridus are rod shaped but slightly carved like this. Not straight rod shape. Okay. So those are based on the shape of the bacteria. Okay. Based on the stain that colors the bacteria, we divide the bacteria into gram positive. Gram is the name of the stain we use, and it came from the scientist who first, discovered that dye. So gram positive and gram negative. Okay. So gram positive bacteria takes purplish, bluish purple color. Okay. So when you will stain, they will take bluish color or purple blue color. So these are gram positive. Okay. Gram negative take reddish or pinkish color. Okay. So these are gram positive, gram negative. Okay. Based on the color. Now, why two different types of bacteria take two different types of color? Because of the structure of the cell membrane. Okay? Just know that the cell wall or cell membrane of gram positive is thinner than the cell wall of gram negative. So which one is thinner?
Gram positive.
Gram positive. Gram negative is thicker, right? Now you tell me, which one should be more resistant to antibiotic?
The thicker wall.
Thicker wall, right? That means negative, right? It is difficult to destroy gram negative bacteria than gram positive. Okay? In one sentence, just know that I have explained how the protein synthesis occurs. The reason is the microorganisms stop the synthesis of protein, right? Microorganisms work by protein synthesis, and antibiotics stop that. So if you can stop the protein synthesis, okay, then microorganisms will not be able to multiply, will not be able to grow. So that's why antibiotics target mainly the protein synthesis. So new bacteria will not be produced. Okay. Viruses are probably the most interesting, thing in biology. So it's amazingly, interesting thing in biology. I'm saying thing I am hesitant to say microorganism. The reason is many scientists, they don't think that viruses are microorganisms. Viruses don't grow. No metabolism is present in virus, right? So, living body must have metabolism, right? To produce ATP. Living cells must have metabolism. Bacteria has nucleus, bacteria has ribosomes, bacteria has metabolism, right? Do protein synthesis, but virus don't do those things. So we cannot consider virus as living organism. But interesting is what? That when virus is not, get close to the living cells, they are like, a piece of non-living thing. So they just sit, do nothing. Okay? They can't grow, they have no metabolism, they can't do anything when they are not in contact or close to the living body. But they become active heavily when they come contact to the living cell or living body. Okay? So they start to multiply very fast when they invade in the living body. So outside of the living body, they are silent, quiet. They don't do anything, like dead thing. Okay? Once they enter into the living cell, they start to multiply. Now, most interesting thing about virus is that they hijack, we can say the word hijack, the genetic materials or function of DNA, RNA of the normal healthy cell, living cell of the body, and start to use that RNA, DNA to produce protein in their way so they can multiply. So that is the most stunning thing, is that they do not use their own material. They enter into the cells and use the cellular DNA and RNA. Make sense? Healthy person's cell DNA and RNA to produce the proteins according to their need. So that's why we say hijack the cellular materials. Now, you see, this is a virus. Virus could be different types. It is very simple. These are DNA or RNA. Okay, and this is the covering formed by protein. Okay, called capsid. Okay, that protein covering. Okay, so that is a virus. So very simple, right? You have DNA and RNA inside it and covered by protein layer, capsid. So you see, nothing else is inside, right? So this is a very simple structure. DNA and RNA could be present in single strand or double strand, so it could be double or single. So this is the basic structure of a virus. See, DNA or RNA inside, and the covering, protein covering, that is the capsid, protein coat. Okay, makes sense? Now, some viruses, they have additional outer layer like this. Okay, that is the membranous envelope, which is phospholipid bilayer, like human membrane, cell membrane, and spikes on the surface. So some virus could be only like this, and some have envelope around them. Okay. Now, how they work? Let me first go over how virus works. Okay. So, this is a cell of your body, okay, and this is the virus. So, virus comes contact to the cell body, okay. This is virus, and this is cell of your body, healthy cell. Virus has entered into the body. It will come contact to the cell body. And what happens sometimes, the cell membrane of this cell and the cell membrane of this virus, they fuse together and it enters into the cell. So, fusion, or we can say fuse of these two cell membranes. That's how some viruses enter into the cell. Another way virus can enter into the cell is by phagocytosis. So this is the cell, and this is the virus. Okay, virus, and this cell will engulf that virus by phagocytosis. So this is phagocytosis. Okay. However, the method is virus try to enter into the living cell. Okay, so that's the goal. Virus will enter into the living cell. Okay. Now you see what happens. This is very interesting. This is the living cell. I have explained you everything here, how the protein is synthesized. So now let's see, the virus has entered into the cell. Okay. So these are virus and RNA or DNA inside them. Okay. So what happens, you see here, these viruses will release their RNA or DNA in the cytoplasm. Okay. After they enter into the cell, they will release their RNA or DNA into the cytoplasm. Okay. Now, let's go back to normal function of the cell. What happens? You remember the messenger RNA, right? Comes, bring the prescription, right? Give it to the ribosomes to produce what? Proteins. What these RNA or DNA will do what? Will come here. Those are from the virus. They'll come here and tell this messenger RNA not to work on the ribosomes here. Okay? We'll push them away. Okay? Let me do the function. Okay? So these viral DNA or RNA will now do what? Will tell the ribosomes, right? Produce protein this way, in a different way. Don't, follow the instruction of the messenger RNA. Follow my instructions. Make sense? So hijack their function. Okay? And now the ribosomes will start to produce the protein. What protein? The protein that needs to produce new viruses. Make sense? So now these ribosomes will make the proteins like this. That will form the capsid. And that is number one. The protein will form the protein of the virus. That means covering of the virus. Okay? Now you see what happens next. This RNA will replicate, will produce new RNA, and this new RNA will enter into those capsids. So now, make sense? So these capsids now got RNA of the virus and become virus. Make sense? Now you got the capsid and viral RNA inside. So you see, this way many new viruses will be produced. So new protein will form the capsid, and replication of this RNA will produce new RNA. They will enter into the capsid and will produce new viruses. Now these new viruses can do what? Produce a chemical that can cause lysis of this body cell.